Well, again, I want to thank the organizers for, one, allowing me to speak in the RNA session, since I'm not going to be speaking about the RNA aspect of this, although I'll introduce that a little bit in tetrahymena. What I really want to focus on today is how we've been using this DNA elimination process to identify novel sort of pathways that regulate and novel functions that regulate genomes. First I want to just give since we haven't heard quite as much about tetrahymena, we've mostly heard so far about the oxytricha and paramecium, just to remind people about how the pathway works in tetrahymena. But first I just want to, talk about what really has interested me in sort of studying tetrahymena has been, and the ciliates in general, is this nuclear dimorphism where they have these two nuclei, and that biology basically reveals a lot of rich things that we've been able to then take advantage of. And so just when we go from one, basically, so one thing is that this nuclear dimorphism has these germline somatic nuclei, which each time have to reprogram a new somatic and germline nucleus. And part of that process is what we've been talking about, is this massive DNA elimination that happens, which in tetrahymena represents about 1/3 of the genome, where while the genome stays intact in the germline genome, during differentiation of the somatic nucleus, you remove about 1/3 of the genome, which has the parts that are eliminated. We refer to the IESs, which I show here with these colored boxes, and they're interspersed within the genes. From an experimental standpoint, what really I like about the process is that, one, it happens it is inducible. We can basically mix populations of cells and take them through development synchronously, and it all takes place in less than a day, that we can go from the prezygotic stages, which is meiosis and genetic exchange, and then the postzygotic stages where this DNA elimination occurs in the differentiation of the new macronucleus. And mostly what I'm going to be telling you are things that are happening in this nuclear differentiation process. We haven't talked too much yet in this session about how are the genomes organized. So we've heard a lot more about paramecium and oxytricha, where many of the IESs, and actually the majority of them, are found within coding regions. Tetrahymena is actually only pretty much exclusively has IESs that are located in the intergenic regions. So actually they may lack the kind of class which is basically the non-maternally controlled IESs, and instead only have the ones that are in the intergenic regions. And so if we just look at a 100 kb region of the MAC genome, it's actually represented in the mite genome as about 150 kilobases. And so if we take this region that I've highlighted here, it has about 27 genes, and in the mite, there's about 16 IESs interspersed in theirs. If we expand that kind of just to this 10 kb region, which is probably one of the best studied regions of the tetrahymena MAC chromosomes, there are it's about a 10 kb region of the MAC genome, or 15 kilobases of the genome, where there's actually 3 IESs, 3 of the best studied, especially the MIS, which I'll talk about, the RIS and the LIS, and there are 4 genes, or the 5th gene right over here. The point I want to make is that the IESs are interspersed between the regions, and there has to be fairly accurate excision to prevent the elimination of otherwise basically controlling sequences or coding sequences. And so this still has to be, even though these have to be quite as precise, and we don't have the conserved TA dinucleotides that we see in paramecium, they still has to be fairly accurate to be able to make sure that you preserve an intact genome. And what I want to tell you and so first the first sort of pathway, which we've heard a lot about in the previous 2 talks, is the way that these IESs are first recognized is by using a small RNA-directed heterochromatin formation. So back in the prezygotic stages, if we go back to think about my earlier slide, here in the micronucleus, there is production of bigenic transcription, which then leads to the production of small RNAs that are, the production of double-stranded RNAs that are then processed by dicer-like in the nucleus, DCL1. That population of small RNAs, which are temporarily controlled and also compartmentalized within the meiotic micronucleus, then are transported to the cytoplasm where they assemble with the Twi protein, which is the one of, I think, about 12 Pwi proteins within tetrahymena. The Twi1 is the one that's exclusively used in this process, where they assemble with the small RNAs. And I'm not showing the selection process in the old macronucleus that Eric Maé introduced very well in paramecium. There's a similar process, although the details are quite, are still, there's lots of things to look at the very difference. But then that goes into the, are transported later into the developing macronuclei, where they somehow recruit, and it's still sort of somewhat mysterious, but they recruit the EZL1 methyltransferase, where it directs both H3K9 and K27 methylation on those IESs, and mark those IESs with heterochromatin formation. And this is a lot of this work, so that a lot of the work on the Twi proteins has been very well worked out by Cosmo Kozuki, who's not here today, and a lot of the work on the methylation process has been worked out by Sean Taverna and Yi-Fan Liu, and we'll hear about Yi-Fan Liu. The one thing I haven't shown here, which since I'll introduce for, I think Yi-Fan is going to talk about actual chromosome breakage. We haven't talked very much about that, but what I'm not showing here, there's one additional process where chromosomes are actually fragmented and new telomere addition happens, and I think we'll get to hear a little bit more about that in the next talk. So part of the process about how these are precisely eliminated comes to how are they recognized, and that's with the small RNAs. But we know that that's not sufficient, as that from work done a long time ago at Minqiao, it was actually shown that while the scan RNAs target this methylation, one, we know that the scan RNAs are not uniformly distributed amongst the IESs. Those IESs can be anywhere from about 300 base pairs to greater than 10 kilobases. So that isn't sufficient information to determine how accurate the elimination is going to be. And so we know from work which really was pioneering work done over 25 years ago from Ron Gudiska and Minqiao Yao, is that there are actually sequences that flank the IESs that determine the accuracy of the elimination. And so just to kind of recount that old data, since there's some of you in the room who weren't even born when these experiments were done, there is that if you basically eliminate one of these controlling sequences, and for the MISs, these were easily recognized as these A5, G5 tracks. There were opposite orientations on each side of the IES. There happened to be two copies of that, about 300 base pairs, that led to alternative rearrangement of the IES, even eliminated 600 or 900 base pairs. But if you remove this copy, you fail to get this excision, and you'll only get the 600 base pair elimination. And another bit of work, if you actually take that controlling sequence and move it farther away, the boundary will move to stay sort of relatively fixed distance, about 50 base pairs away from the position of that sequence. That's an important experiment because it says that actually the sequence actually at the excision junction in the wild type cell isn't what's important for defining where those sequences are, but it's really the distance from this controlling sequence. And so for the work stood there for a long time because, one, the MIS was the only IS originally identified that had this A5, G5 sequences. Other eliminated sequences that were studied in various labs, including Ron Perlman's lab, who's here, and Kathy Carr's lab, showed that there were similarly positioned controlling sequences flanking the IESs, but none of them had any identity to one another that we could really understand what those sequences were. And that brings us to the work that I really want to talk about today, really the groundbreaking work was done by a graduate student in my lab, Christine Carr, as she was studying identified the Lia3 protein. And I'll tell you about mostly on some new work from a very talented undergraduate in my lab, Vita Jaspin, who studied a homolog of that protein called Lia3. And so I just want to point out that these two students really made all the work that I'm going to tell you about possible. And what I'm going to tell you is there are proteins that we've identified that they're interacting with these sequences that are controlling the accuracy elimination. So what are these proteins? Well, Lia3 and their Lia3-like proteins 1, 2, and 3 appear to be tetrahymena specific proteins. They do share about 100 amino acid region in common, which I show aligned here. And they range either from about 275 to about 400 amino acids in size. What really brought us interested in Lia3 in the first place, we identified it in a screen for proteins that were expressed only during development, which is sort of shown here. They were not expressed in growth or starvation, but their expression of all of these peaked about 8 hours in the conjugation, which is in that post-zygotic stage just prior to the DNA elimination. But other than that, we knew nothing about these, and when Christine originally knocked out Lia3 and Vita knocked out LTL1, which I'll show here, the data for here, we found that actually the boundaries of IESs were affected, okay? So the knockout of Lia3 was recently published, so I won't show that, but Vita knocked out the LTL1 protein, which is the data just shown here, that if we replace the LTL1 gene with a selectable cassette, we no longer in the mutant strains have any copies of the wild type, and only see the mutant allele. When Christine looked at so I'm going to basically kind of combine the two data of these, didn't go back and forth between the study of these two different proteins because they are actually very similar stories, and we'll talk about what the differences are. But so in the case of the knockout of Lia3, what was different than many of the proteins that we've studied that are required for DNA elimination, most of the time we heard from Rachel Howard Till-like, if you basically have many mutants that fail to do IS excision, they arrest in development. Mutants of Lia3 actually produce progeny. There was somewhat decrease in the actual viable production, but actually they produce viable progeny. The same with the knockout of Lia5. Basically, actually, while Lia3 showed a decrease of about 30% progeny production, actually mutants in LTL1 actually produce progeny almost as well as wild type. And when Christine was first looking at Lia3, it actually stymied us for a while trying to figure out what this was doing until we really started looking very carefully at the IS excision. So basically, this is a particular IS that's actually flanked by these A5G5 tracks. This is about a 9.2 kb ISs, and you can see in the parental lines or in wild type progeny, it has very accurate elimination where we see a uniform product in all the individual progeny. But in the progeny of Lia3 mutants, we basically saw this very heterogeneous boundary formation. So basically it says that no longer is the excision of this IES accurate. And this was actually true of, now at this point we had my genome sequence, so we were able to go through and actually find other IESs that have these A5G5 sequences flanking them, including the MIS, which I'm not showing here, but where basically in normally wild type cells or in the parents or in progeny, there's very accurate excision of all of these. So you see a single product, where in the progeny of the Lia3 mutants, now you see very heterogeneous boundary formation. And this was true for all the ISs that had flanking A5G5 sequences in Lia3 mutants, but for those that were not affected basically didn't have these controlling sequences. So it appeared that Lia3 was specifically regulating the precise elimination of the class of ISs that are flanked by these A5G5 controlling sequences. Actually, we sent these strains to Ming-Chao Yao's lab, and a talented a grad student in his lab has actually looked, and there appears to be about 100 of A5G5 linked ISs within the genome that are regulated by this protein. Similar thing, Vita Jasmine found with LTL1 is that basically when she made the knockouts, and actually now in this case with the Lia3, we originally looked for ISs that had these A5G5 sequences, and all of the ones that had these flanking sequences were regulated by LTL1, by Lia3. With Vita, she actually took 200 kb regions in the MAC genome, which had roughly around 30 IESs, and did an unbiased screen, and basically found that there were 19 that were unaffected, where there were 8 that either showed aberrant or altered excision in Lia3. And all of these were different ones and did not contain the A5G5 tracks. What do we mean by aberrant versus excision? Well, those that had aberrant excision, we call those as basically, she described those as ones that had many different boundaries in the progeny of the LTL1 mutants, where those with altered excision primarily had a major rearranged form. What this may mean is actually there are some type of additional controlling sequences that reside that if you knock out one of these, there are actually sort of backups that kick in. And maybe that's one reason why the progeny production is still quite good in these cells. So the hypothesis then, of course, is that Lia3 and LTL1 are binding to these controlling sequences to regulate the accuracy of elimination. Christine and Vida expressed these proteins in E. coli and purified the proteins. And first I'm going to show you what to work with Christine's on Lia3. So she purified the protein and looked with IES flanking probes to look for the whether these are able to bind. And initially she tried very hard to actually so the MIS is a sequence that has this A5G5 sequence, and she tried very hard with various types of double-stranded probes and basically never saw any binding to Lia3. However, when she took initially we thought was single-stranded M1 and did the binding, so here is just a free probe, and basically here we see, when we add the Lia3 protein, now we see a shift, so it very specifically bound this. But what became really apparent is actually the free single-stranded probe should be running on this gel down here, but even the free probe was running at an aberrant size. So what is this aberrant size? Well, this A5G5 containing flanking probe has G5 tracks, and it's not surprising that this can form a basically non-canonical DNA structure, which is a 4-stranded structure called a G quadruplex. So if you're not familiar with these, they're actually 4-stranded structures that are coordinated by a potassium ion or basically a salt of a specific size, and they form they can either form parallel or anti-parallel. Actually, some of these were first shown in vivo to be able to form on tetrahymidal telomeres using antibody probes, and also in other ciliates that have been shown to be able to form on telomeres and other organisms as well. But it's been, for a long time, we've not really known, can these actually form in genomes in vivo? And so basically to actually show this was a G quadruplex, Christine used circular dichroism spectroscopy, which basically G quadruplexes have a very unique spectra, and this actually showed that this was a parallel G quadruplexes. Telomeres form mixed G quadruplexes, and they were specifically binding to that sequence. And that's shown sort of further here, is that when we repurify the protein, it doesn't bind the single-stranded probe, and we can keep the M single-stranded by just actually using lithium instead of potassium, which isn't able to coordinate the G quadruplex structure. If we form the G quadruplex structure, adding additional Lia3 protein bound specifically to that A5G5 sequence. But if we purify a random protein in the same way, we see no binding, so it's not just something in the E. coli extract, just to be sure. And just to show that it really is specific for that, we tried to compete away either with the G4 quadruplex, and we can compete away binding, but if we mutate and replace that G with either 1, or in this case 3 Gs, where the G quadruplex can't form, it's unable to combine. So it's not the sequence, but really is that structure that Lia3 is binding to specifically. And this was very exciting for us because it really suggests that we can make a mutant basically see that it's affecting the sequences in the genome that basically have these G5 tracks. We know that those are the controlling sequence, and in vitro we see that basically Lia3 binds when that is only in the G quadruplex structure. So it really gives us strong evidence that these non-canonical structures can actually form a genome and have regulatory structures. Now, we've been trying to figure out, so now what does LTL1 bind? Okay, so LTL1 does not have these G tracks flanking. The IESs that are affected by LTL1 do not contain these G tracks. They actually look like very AT-rich sequences that it's really hard to identify them from any other non-coding sequences. What Vita was able to do using probes from what we think is the controlling regions of the IESs, although we don't know that for sure, and I'll come back to that in a minute, is that it didn't bind single-stranded, but it bound double-stranded DNA, or even the G quadruplex as well that's not regulated. And when basically she was looking to see what's the affinity of these sequences. So Lia3 bound to the G quadruplex when it was at about an affinity of about 100 nanomolar. Didn't matter whether we looked using double-stranded probes to two IESs that are actually regulated by LTL1 or the G quadruplex, we basically saw a similar affinity of about 350 nanomolar, so not as strong as Lia3 binds, and basically there was no really distinguishing ability for LTL1 to bind this. So this made us actually wonder, have we really actually identified the correct region that LTL1 is binding? So we took, this is what we refer to as IES D, and Vita basically did sort of deletion analysis to see, well, where is the controlling sequence for this? So in the case of the M element and other A5, G5 flanking IESs, it's about 50 base pairs away from the actual boundaries. So basically she made deletions either into about 115 base pairs or made these internal deletions, and what she was able to show for one, basically she was able to delete all the way to 115 and get very efficient elimination. All these mutants actually rearrange very efficiently. When she made these internal deletions, which is not actually, it's shown here, which it's hard to see down here, the clock is in the way for there, but so we can see that the boundaries shift to actually move to stay a fixed distance from this controlling sequence. So what this says, actually, like the other IESs that have been identified, there was something in the controlling sequence that works in the distance to determine where the actual sequence, but actually it appears that the sequence is somewhere between about -75 and 115. In recent, the last sort of preliminary data really suggests that actually there is a higher affinity for the protein than this, but it doesn't clearly have as strong as a distinguishing sort of affinity for that sequence that I would say that we've really identified that it's binding that just as double-stranded DNA, and I'm still sort of conjecture whether there may be some additional structure that they may bind. So I said that Lia3 and LTL1 are novel proteins. In trying to understand where these might come from and what they actually represent, Vita did structural prediction analysis, and they actually, if you do this analysis, in addition to basically finding each other in sort of an unbiased analysis, it finds that this is basically its highest prediction is over about 24% coverage, is similarly to DNA binding motifs within TC3 DNA binding domains of these transposase proteins. So I think it's likely that these represent basically DNA binding proteins that were domesticated from transposase, but they've diverged in such that it's really hard to recognize that. We can only see in the structure, in the function. So in this conserved region, we wanted to know, is this region important for binding? So we basically looked at these two really conserved regions, this GRG or GKG, or this IYI motif, and basically made alanine substitution mutations. And Angus Tolan, who was a med student who was doing a summer rotation in my lab, basically was able to show that while Lia3 mutations, either having the GRG or the IYI mutation had basically no effect on binding, but specifically mutating this GRG basically abrogated binding of Lia3. Vita wanted to say, are these, though, regions actually functionally basically interchangeable between these two proteins? So basically she made chimeras where she swapped the LTL1 Lia3 conserved domains, and then basically expressed these off an inducible promoter, and these are put into the Lia3 mutant cells, where she expresses just the intact Lia3. Without induction of the expression, she sees very aberrant rearrangement, but you can see is that expression of the intact protein restores very precise elimination. However, neither the chimeras expressed within these cells were able to rescue the mutant. We can do a similar thing where we've rescued the LTL1 mutant with LTL1. We haven't yet done the chimeric experiments to see, but it appears that they aren't exactly functionally interchangeable even though they have conserved, so there is still some functional diversification of that. So just to summarize, we've identified these two proteins, Lia3 and its homolog LTL1, that regulate unique subsets of the eliminated sequences. They bind different sequences. We know Lia3 specifically binds G4 sequence, G4 DNA. We think this actually is one of the first really clear examples where we have a protein that binds to a G quadruplex structure in vitro that actually regulates that same sequence in vivo, and I think this is actually one of the best evidence there is for there's actually G quadruplexes can form in vivo. Those conserved regions aren't functionally identical, and they may have been evolved from transposases. And so with that, I'll basically stop to thank the people. Really, this work was pioneered by Christine, and she did a lot of work during her thesis work that actually showed that Lia3 binds to G quadruplex, and Vita Jaspin, who just graduated with her undergraduate degree. She's going off to New York Med School, but she's been working really hard over the last 3 years. Angus Tolan did the mutational analysis, and Marta Tay, an undergraduate, is continuing this work in my lab. And these are other members of the lab, and Joyce, I will say, helped a little bit with Vita's project last summer. And I'll stop and take any questions.
Thank you. We have time for a few questions.
Sorry, Jack, I don't quite remember how you envision the G quadruplex forming, because it actually requires 4 stretches of Gs,
so
right?
I put this in here. I didn't know if I'd have time to get to it, because, I mean, that is still a conundrum to us. How do you form a G quadruplex when you only have 2 G tracks here and here? And one possibility we think is actually the non-genic transcription, which we know is occur, and this is RT-PCR showing that there is non-genic transcription. It could be that these are an RNA-DNA hybrid. So maybe I do belong in the RNA section, but we just don't have the data to show that. And we do know that Lia3 binds an RNA quadruplex as well as it binds a DNA quadruplex, or we can compete away with that. It may also be that these G quadruplexes form, there is around the DNA replication that happens before. It could be sister chromatids are held together, and they're actually the 2 IESs are coordinately excised, and it's actually replication which allows these to form, but we're still trying to distinguish those. Jeff?
I was just wondering if you relax the stringency and you look for other homologs of these proteins, do you have any hints about where they might have come from? Like any transposon or something like that?
Yeah, I mean, like, the best has been this sort of structural prediction. Things, ciliates are so far away from everything else. And I'm not an expert in that type of analysis. I think somebody who is better at that type of prediction may be, but it's likely some ancient sort of, these probably been in the genome fixed for a long time, and so I think that we would like to know that, but we can't be for certain. Bob?
Hi, Doug. So I don't know how to phrase this question, but it seems like this involvement of special proteins for only a very small subset of the IESs kind of abrogates the whole beauty of the scan RNA model, which is kind of a universal model for how IESs are recognized and excised. And you've got so far 2 different, or a few proteins, but you've got such a diversity of IESs in only one species. And then if you consider the fact that in other closely related species, the IESs are in completely different locations, you've got a whole mess here. So how can you...
How can I reconcile, so what I guess I would say is that, for one, we think it's important, at least in tetrahymena, that you have these boundary controlling regions because of the span of sequence that these are able to eliminate. And we know at least that LTL1 regulates the adjacent IES to what Lia3 does. And basically, if you don't have something that basically organizes the genome in a way that that prevents basically the end of one actually spanning and deleting a gene. Yeah. I think these are actually needed because of the diversity and actually the size that can be eliminated by the scan RNA process. Yeah. The other thing is that my guess is that the proteins that regulate these have basically expanded, basically the transposons that actually were able to basically have their proteins domesticated and regulate them, because they probably were originally sort of the binding sequences of the transposase that land there. Those were probably those IESs that spread and became IESs, and there's probably a handful of whatever IESs that really have derived the bulk of the IESs in tetrahymena. Or once you originally get these, you can then insert more insertions into the IESs that are there. And that's maybe why we're seeing that aberrant elimination or the altered elimination boundaries, the precise ones in the LTL1 mutant. So that wasn't a very articulate way of saying it, but so Larry and you, yeah, I'll answer them quickly.
I guess one thing I was struck by today is there accuracy really necessary for most IESs in tetrahymena? It seems like you can get away with a lot of slop in the system. Perhaps there are a few IESs that have to be taken out accurately, but maybe it doesn't matter for most of them.
In the lab, that's true, but whether they get sick or basically if you actually in the environment, we don't know. But that's it's a good question. Mireille?
It's a quick question about Lia3. You said it was more specific for a structure rather than a sequence. I was just wondering about telomeres. So do you have evidence that it can bind to telomeres?
We tried to compete with a telomere oligo, and that didn't bind, but they tend to make mixed quadruplexes and more likely anti-parallel, so actually it doesn't look.
All right, okay.
And we still wonder whether LTL1 doesn't bind some other structure we haven't identified yet.
Okay.
The time for the last speaker for this